exciting day with lots of wonderful presentations. And I am encouraging people to move forward. We are going to have two brief presentations from two of the proteomics research groups, and then look forward to discussion among us, both ideas for new projects, new research studies, ways either that the research groups can work together, and also looking for new members for our research groups. So we hope you'll be open to volunteering. So we'll start with Lindsay. Are you starting? Lindsay Pino, representing the Proteomics Standards Research Group.
Thanks, Sue. A couple of you are members of the sPRG, so this will be a little bit of a review, but wanted to share with you what we've been working on in the sPRG for the past couple of years now, including some fun pictures from right here in Minneapolis. I am part of the sPRG, conflict of interest. I'm the co-founder and the CTO of a small biotech startup group, and I have a collaborative research agreement with Bruker Daltonics. None of that has come into play in the research that I'm going to be talking about today. So in the sPRG, kind of the main overarching thing we're thinking about is quantification and harmonizing or unifying benchmarking quantification across all of the various different platforms, both hardware and software, that mass spectrometry cores, CROs, laboratories, research laboratories in academia and industry are using. So to illustrate this, I'm going to use my personal favorite figure, because I made it. If we imagine the yeast proteome and measure the abundances of all of the proteins in the yeast proteome, we can plot on the x-axis how many protein copies per cell. And then we can see this distribution where on the far right, some proteins have very, very high copy number. On the far left, some proteins have very low copy number, but the majority fall somewhere in between. In an ideal world, we would measure all of these with our proteomics techniques, but that's not the case. I think most of the proteomics techniques that people are talking about, especially if you're trying to measure yeast, you're going to be using a mass spectrometer. And a mass spectrometer can't measure all of those proteins that are present in the yeast proteome. Specifically, it's left censored, so you see a shift. This blue distribution moves over to the right a little bit. Low abundance proteins are hard for us to detect. That's what's shown here in blue. If we ask another question, though, how many of those things we're detecting can we quantify? You see another subset, another shift farther to the right. So not only is it hard to detect low abundance things, it's also hard to quantify low abundance things. And if you've looked at a chromatogram, you know that mass spectrometry is not inherently quantitative. So this is showing three samples, the three samples that the sPRG has put together, one peptide from one protein in those samples. And you can see just visually, whether you're a mass spectrometrist or not, going from left to right, it looks like there is an increase in the peak area, an increase, what we would like to think, in the abundance of this peptide over these three samples. And that's what we would like to see, but mass spectrometry is not inherently quantitative. If I were to take these three samples and leave them out on my bench for a week and then try to measure them again, this might look different. If I take the same three samples and put them on one hardware, one instrument next to another, we might see a slightly different answer. So what we want to do with the sPRG is come up with a way that we can describe the precision and the accuracy for quantitative proteomics measurements. So the precision in our field is usually described as the percent coefficient of variation, the percent CV. If you made the same measurement multiple times and then you calculated the mean and the standard deviation of that measurement across all of the times that you've made it, you can come up with this percent CV measure. That's great. It gives you an idea of how precise, how reproducible your measurement is, but it actually doesn't tell you anything about how accurate that measurement is. If you think back to like your analytical chemistry and the classic bullseye picture, right? And it's like all of the shots are kind of clustered together, but they're not on the bullseye. That's very precise and not accurate. So accuracy in the field, people have started using this idea of measured ratio between two or more samples, where you know what the ratio value should be. So if you take sample 1 and you take sample 2 and now you compare the ratio between those two samples, you get an idea of how accurate your quantification is. And to our knowledge, to the working group's knowledge, we don't have any commercially available standards like this. There are other mixtures out there, some that have come out of ABRF working groups, like the UPS, Universal Protein Standard Mix, that can capture some quantitative accuracy and some detection benchmark values, but for 15 to maybe 400 proteins. What we wanted to do was go forth into this highly unbiased, measure everything in one sample approach that a lot of proteomics users and groups are starting to get at. So we need this scalable quantitative standards that we can compare different hardware. The vendors are always saying that they have the best detections, they have the best accuracy, blah, 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 blah. You want to see it in your own hands. You want to see it in your own lab, especially even if you have different operators, if you're comparing different softwares, and you're trying to get an understanding of what is happening to your data. So we want a quantitative system suitability standards that we can measure, for example, the same hardware over time. So kind of that rigor, reproducibility, transparency that we were talking about earlier. If you made the same sample and measured it over the lifetime of your instrument, you can get an idea of the performance of your instrument with quantitative benchmarks, not just the number of helipeptides you've detected. We want to go between systems, between sites, and between softwares. And then finally, we wanted a standard that might be hard for the software vendors to analyze. We don't want it to be easy for them. We want them to struggle. If it's easy and straightforward, they've already over-optimized on those kind of samples, so we wanted to kind of throw them a curveball. So in the sPRG study group, we actually have like 50% attendance here. Pretty good, yeah. We wanted to design this standard material. So the head of the working group is Ben Neely. He works at NIST, so he obviously had a preference for using NIST materials. We picked 3 proteomes, human liver, cow liver, and fish, and prepped big batch, each individual, a big batch of digests. I did all of these with S-Trap meaty preps, multiple MIDI preps that I had to then combine. And I learned a lot of interesting things along the way, which we could talk about, like how much protein is in liver. It's not a one-to-one weight number, things I learned. So we had these 3 digested proteomes, and then we got them all clean, so now we have these 3 pure species proteomes, and then we mix them at known ratios. So we know what the answer is because I literally sat there with a pipette and made those ratios. Now we can test the hardware, the software, and see if they got those known ratios that we made. So we've got these sPRG peptides a couple years ago that I rushed to get out before ASMS in 2022 came literally right here, and we did a couple tests. So we checked, for example, just the empirical detections from these NIST-based materials. And remember, we don't actually care how many detections we're getting from this. It's not a detection measuring test. The idea is the ratios. And intentionally, we designed these materials to have at least some tricky overlaps, especially between the human and the cow liver. A lot of those proteins are evolutionarily conserved, so it's hard for a software to determine, impossible, for some proteins for a software to determine if they came from one species or the other.
Lindsay, I've got to interrupt there. Are you saying that there's only that much overlap? Are you talking about 100% homology between?
These are just like peptides. So if you take a peptide and you measure it like in one of these bar charts, and then you just ask, is that same sequence in one of the other bar charts? Not in this analysis that we did here, but you can start to see exactly that when we get to the quantitative accuracy plots. You can start to see the stripes that aren't changing that are supposed to change. So that's the spoiler for kind of this next thing, right? So we like made all of these aliquots, speed vac down all of these aliquots, got them all labeled by hand. Speed backed them down in batches, walked around with some sketchy tubes with white substance, and got them ready for distribution in little dime bags over at the convention center. And then I walked around and I just handed out bags of tubes to whoever wanted to take these 3 samples. They don't know anything besides it's a human, cow, fish abomination, and they're going to measure each one of these 3 samples on their instrument, analyze the data, and just send us the results. They don't know what the ratio is supposed to be. They're just supposed to guess based on what they measured. So now we have this bake-off. We ended up having 12 sites submit 15 data sets. That means a couple of the sites submitted multiple data sets. One of them was comparing 2 gradients. They wanted to do the same samples on a short gradient versus a long gradient. And a couple other people had different ways of running data-independent acquisition methods. We first start just by looking at some of the peptide detections. So this is each one of the submissions going up on the y-axis, and then we're just going to show you how many peptides they detected for each one. So you see for some of these mixtures, this is actually a pretty hard problem. There's a lot of peptides in there, but detecting them and calling that detection is pretty tricky when you're using some methods or when you're using some software. But that is vastly different for the different sites. So different submissions have vastly different, even just numbers of detections. So you can start to see some of these sites that had submitted multiple data sets. One of them was comparing different ways of doing libraries, using a predicted library versus an empirical, a gas phase fractionated library. I wonder who that was. And then another one who was doing a short gradient versus a long gradient. So you can start to see some trends here that the analytical length of your chromatography gradient might not actually make a huge difference for your detections, but the software approach that you're using, the analysis approach, does make a pretty big difference. So then we start to make these plots where we have the abundance on the x-axis. So more abundant things are on the right, less abundant things are on the left. And then on the y-axis is the ratio. So we need these 3 ratios. And there's the human, the trout, and the bovine. And remember, we didn't tell people what the ratios were. So they just sent us numbers. So we can start to plot these. So in the green, in the green, we are showing the cow. In the kind of purpley blue is the fish, the trout, and then in the orange is the human. So in the first site, you can start to see high abundance things, easy to measure, you get the ratio accurate, and it's right. And then low abundance things, harder to measure, you see spread away from what, like visually you can tell is what the ratio value is. So we can do this for all the sites and all of their various analysis approaches. You start to see some interesting trends going on here. This is kind of where we're at, this is everyone did their own data acquisition, everyone did their own data analysis, and then gave us those results. So some of the next things we're going to do is take all this raw data and shove it through the same pipeline, analyze it all the same way, and try to strip some of that analysis variation out of it and see what the method is.
On those plots, but that's not one that, for example, I'm used to seeing.
Yes.
Would you point out what you would consider the best result there?
Man, that is pretty hard to say. The best result, what you would want to see, is a very thin line like this plot has that goes thin line all the way across. Oh, do we have a pointer? There we go. So these on the right-hand side of these plots where it's high abundance proteins, you kind of get just like all the points piled up on a horizontal line. That's what you want to see. You want to see lots of points closely clustered on a horizontal line. You don't want to see stripes vertically. You only want to see stripes horizontally. So this is a fun one. You want to see lots of points. So this one's got a lot of points, but it's very wide distribution. So pretty high variance in the quantitative accuracy. Can you imagine if you were trying to call a differential fold change in this, and it should just be 3 lines, but you've got such a wide variance in the quantitative accuracy. It's hard to say what is good. Like this plot over here is missing a lot of detections. You can see it doesn't really have a lot of the high abundance proteins for the human or the bovine. Maybe having trouble calling the species in those scenarios. And a lot of the human points that should be up higher because they were changing are kind of down in the unchanging fold change of 0 line here, right? So this line right in the middle at 0 is all the peptides, the species that should not change at all between the samples. And this approach here is saying a whole bunch of things aren't changing when we know they should be changing, because I literally put different amounts of that protein in the sample.
So, if I remember, the number one had very few peptides, and yet here
Less
it looks
detections.
like it has the same number of points as number 7, or even more points than number 7.
1 in 7.
The most, or second most
Yeah.
peptides. So how does that work out?
Just the density of the plotting. If we decrease the alpha on these points so that they're more transparent, it would probably be more clear. That's a good suggestion. When we're continuing to do these data analyses, we can add an N on the top of these plots to make that more clear. And we'll be making some box and whisker plots so that visually you can see the spread a lot better.
I agree with Sue. I mean, I've analyzed with the mixed database of bovine and human, and there's a lot of overlap.
Yeah, and you can.
Doesn't fit what you're showing. I don't understand.
Well, this is probably only going to selectively be showing you things that aren't overlap, right? And you can,
That other one, the Venn diagrams.
yeah, I mean, that one's just kind of PSMs, right? This was in the very, very beginning here, right?
That doesn't make sense.
I would agree that's a very small overlap for the.
Yeah, we have to repeat this because this is obviously DDA and PSMs, and it's not the DIA that we ended up going with. The idea here is just, what I'm not showing you is we also evaluated a lot of other proteomes to see how they looked. So tomato leaves, apple leaves, forget what else.
Based on your experience, what would be the ideal mixture? Would it be this here, or would you have
Yes.
a plant?
No, I wouldn't want to plant. Plant leaves actually didn't have a lot of peptides and proteins. It was just overly dominated by a few select, yeah.
I think we just need to look at that again.
Well, yeah, it's DDA. Of course we're looking at it again.
Well, the DDA is still, we get lots of overlap with DDA.
Yeah.
I think we need to reinvestigate that.
But on that plot there, would the ones that have, like the, I don't know if it's supposed to be red or orange, that run over into the blue and green, that's showing overlap, isn't it?
That's probably peptides that are not being they're being conflated, right? So if you imagine you have a peptide from a protein that's coming from the human and a peptide from the same sequence coming from the bovine, when you mix them, even if they're at different ratios, as long as those ratios add up to 100, and one of these is not changing, then they're just complementary, and they're just going to cancel each other out and show no change.
For example, some of those would have been generated with only what in scaffold you would call exclusive peptides. So I don't see how those plots, if anyone separated them, how anything can overlap. I mean,
So-
we don't need to go into it now, but.
Do you want, I can answer that.
Go for it, scaffold.
Which is that a peptide can be unique, but the underlying data that you're integrating is going to be conflated potentially if there's interference. So if you have a peptide that's being interfered with a similar, or the similar peptide of an alternate species, and you're integrating the entire precursor area, for example, then you might not, your precursor quant might not be representative of the detection. So the detection might be uniquely assigned to one species, but the quant might come from multiple species.
Because it's, yeah. Yeah, I think these questions bring up some interesting points to put in the discussion for how to kind of interpret these types of plots and explain how any point that is not purple that falls on this line of 0 is a mistake at some level.
I mean, for example, when I do DIA, if there is a peptide that's shared across species or across proteins, I don't count it.
Do you do that within the software or after the software has given you the result? I don't think every software does that.
I know, but I know which one is mine, and I'm showing overlap there.
Then then it is like Brian's saying. Even if you're only using those peptides, I think there's not a small number in this particular because it's liver, so it's also a limited proteome. We're not looking at the whole human proteome and the whole cow proteome.
Lindsay, just a question on the experimental setup. So you had
Yes.
3 tubes, and you're doing the ratios between the 3 tubes. Is that right?
Yes.
So there's 3 tubes with different ratios, and people were just supposed to do whatever intensity-based measurement they wanted, or did people also do?
Whatever label-free quant they wanted. They could have tried to do TMT. I don't think anybody submitted TMT.
Yeah, there's no reason why.
Yeah.
It's just getting 3 tubes of peptides. You can do anything you
Yep.
want with them for quant.
No, I just wanted to know
Yeah.
what you got back.
Yeah.
We did get some spectrum counting data.
They're already mixed, but I could imagine that you could do TMT because we want the ratio between them,
Right.
not the ratio within them.
Label one sample and then the other, so
Yeah.
sample A would be labeled with 114 and sample B 115 and sample C 160.
And then you'd be comparing the ratios. I think that would work.
No one did that.
Was it a good idea?
Yes, I have a ton of tubes.
Livers? I mean, if you really want to, I mean, I understand you want to make it challenging, but
Yep.
you're really making it overly challenging in some ways.
I think that's good.
I mean, the goal was to make it,
Oop.
the goal was to make it challenging to an extent, right? We wanted a population of peptides that we knew were not going to change the trout, and we made it so that those were the same one after another, in all of the ratios. And of the remainder, what, 50%, we allowed a portion of that to be human and a portion of that to be bovine in each of the different samples. So the question was, could you quantify the differences? And knowing that there would be overlap in peptide sequences, and say, could you separate those out from everything else? So if you came at the problem knowing that bovine and human are going to be different, and you're expected to tell the difference between bovine and human, you should be doing quant on just the peptides that are unique to each one of those. And that seems fair, right? But it gives us a way to have a controlled environment where most of the peptides are not actually changing. Only some of the peptides are actually changing, and that means that it reflects more like a real biological experiment.
Did you provide the database?
We did
Yep.
provide the database.
But
Yeah,
you're
the FASTAs are
biasing,
provided.
right, based on your trypsin digest for certain peptides, regardless of it's from bovine or from human. So you're gonna get, 2 peptides for quant, and they're gonna be the same 2 peptides regardless, because that's gonna be the ones that are gonna come out by trypsin digest.
What do you mean? I don't understand what you're saying.
I'm saying, like, you're really overly biasing this experiment, is what I'm trying to say, by having like 2 liver samples, because you can see that fish is super tight.
Well,
I understand what you're saying.
exactly.
I
I mean,
just
the fish is supposed to be
kind
sourced.
of, I understand. but
Yeah.
It's like,
Because it's the thing that's not changing.
I understand that. Just
It's much harder to call a far ratio than a
sure.
close ratio, right? It's easy to call 1 to 1 to 1 to 1, right? It's hard to call 1 to 10 accurate, right? It's hard to call 1 to 20 accurate. And so when you see that scatter in the human and in the bovine, that's where you're looking at, is it 1 to 5 or is it 1 to 6, right? That variation that you see in the ratio. That's where it's a little bit more difficult to make that call. Or if it's like 1 to 20, it can be a little bit more difficult to make that call than 1 to 1 to 1 to 1. Does that make sense?
yeah, it makes sense. It's just a hard.
How would you think it should be set up?
I'll throw yeast in there instead of both livers. I mean, I get what
Too
you're
easy.
doing here. I understand what you're doing, but
Yeah.
just to get like, can we get stronger ratios between them?
But the goal is not to get stronger ratios. The goal is to break it. The goal is to break this in as many ways as we can. Because the easy one that everybody does is you mix HeLa and yeast and E. coli, right? Like, that's so boring. And it's not really a good experiment either, because humans so much more complex than E. coli.
And the amount of protein changes in each sample because you're diluting very different types of species with each other, right? And so that's where, like,
Yep.
it can be really difficult to interpret one sample versus another because of all those subtle, like all those really big global changes. We wanted something that was a little bit more subtle that reflected what a biological sample would actually look like. We wanted it to be hard. We didn't want it to be easy.
So did you send out the digest or the entire protein?
The digested ratios, all people had to do, this one this particular standards group, we didn't want to test your sample prep. Other groups have already done that in the past. We wanted to have a standard that you would go and buy, and you would now use this on your system to assess the instrument quality over time, right? If you're seeing detections stay the same on your instrument, but your quantification is going all over the place, there's something wrong with your system that's not being diagnosed by just running a DDA HeLa sample, right?
So I have a different question. So what are you evaluating based on finish? Because now that you've discussed it a little bit, I would, sorry, I'm asking what is being evaluated by the fish data, because now that I see this, it looks like number 3, if I would actually like to see just the fish data by itself to see how that's distributed.
Yeah.
And that tells me a lot about what I should expect from
Yeah, we can do that. We can slice this out and see how tight these distributions are. These are pretty hot off the presses. not too long ago, these were generated. And slicing each of these out and making box plots for each one of the ratios in each one of the samples is like a pretty classic thing people do with this data. And you can, yeah, 3 is pretty easy for this particular approach and this particular analysis to call. Interesting that there's no high abundant proteins from the human or the bovine, but yeah, trout. This particular approach loved the trout.
This is kind of dismaying, because this scatter is rather, like the lower left is acceptable with some outliers, but man, that's a lot of scatter.
So you can read this a few different ways, but I actually think that this represents something that is kind of nice to see. Every one of these people got essentially the right answer, right? When we asked the question, what was the ratio between the human and the cow liver samples? They got the right answer, or at least close enough to the right answer that we were calling it good, right? I think answering this particular question was a hard question. And while there is scatter in the data, the actual end result, and this isn't necessarily the best way to represent this because we can't see how much density was behind it,
Yeah.
right? But the data actually came out much cleaner than it had any right to be. And I think the answer is that there is much more consistency amongst labs in the proteomics community than there was 10 years ago in terms of quantitative accuracy.
But you're talking about aggregate data getting the right answer. How about what's the range of individual ratios within the set? Because if you don't want, if you're looking at a data set, you're really not interested in aggregate. You're interested in what the individual proteins would be telling you as an investigator. So if that is a really wide distribution, but they come back to the same ratio in the aggregate, it's still not good.
There are ways that you could pick this question apart and come to a more negative answer, and I think that yes, you can do that. What I would say is, in general, people work pretty good at getting the right answer, and that this way of representing it isn't necessarily the cleanest way of representing how consistent the data actually ended up being.
But it would be neat to look at for protein ratios.
So, and certainly some labs would have more variability than other labs, and certainly if you made more calls, if you called more proteins, then you had more variability in those protein measurements, right? So, definitely.
Well, it's going to be a function of signal noise.
Right, so
It should be.
as you're trying to detect further into the graphs, there's more variability at those lower measurements, right? So in those cases, calling more things, you would see more variability. And so, and I think much of what you see in terms of variability in the spread of those measurements is more of a function of how many proteins did they actually make the call on versus the quality of the measurements themselves. And again, I don't think that this is well represented because inside of each one of those red blobs, you're looking at thousands
was intensity and ratio or probability of getting a ratio, and that would be a really great curve to display here.
Yeah, I think we have some more ways we need to pick this data.
But I mean, that's.
I think that's definitely true. I think maybe something to remember is we're the standards group, not the information group, actually. I think these are all great ideas. The goals, though, of this particular project was to build a physical standard so that we can compare these things and do all of the great ideas that we have here, but to make the standard, not necessarily to make all the algorithms.
Paul's point is really important, that most
But that's all I got.
people want to use the standard to look at protein ratios across the samples, not the aggregate of the species.
In a sense, the data set or the sample set gives you the ability to say, was this peptide accurately measured in a very difficult circumstance, right? And to be clear, this is much more difficult than a normal proteomic experiment. So
Intentionally.
it's not surprising that there is some significant variability in these measurements. But it gives us kind of a hopeful idea for that we are able to make reasonable measurements, at least in the aggregate, for each of these different things, right? So again, each one of those points is a different peptide, not a different protein. Already, when we're trying to do proteomics, we're typically aggregating that data anyway. And so there are different ways that you could think of this and separate it out. So you can come away with a negative answer to this, but I actually I do see a positive answer out of this, which is that there is a lot more consistency in the field than in being able to do basic mass spectrometry, and not just basic mass spectrometry, but actually the informatics to back it up, right? 10 years ago, or 15 years ago, there was a study by the Coupeau group talking about how even if everyone analyzed or collected the data the same way, their analysis was such a problem that there was no good solution from one sample to the next, or one lab to the next. They basically all said completely different answers. And that you had to analyze data their specific way to get consistent results. And even though all the quality of the underlying data is actually pretty good, here we're showing that all of the informatics analysis is actually pretty good as well.
I'm just wondering, is the plan here, for instance, these 12 sites, to then, like, maybe a few months from now, do the experiment again, and sort of cluster, see like how reproducible that same site is to themselves, like, over time? That seems like that's what you're going for here, right? Like longitudinal.
Well, our goal is actually to commercialize a physical sample. So I think our next big steps are to get these samples somewhere where you can purchase them. I still have some in my desk drawer if you want to get in touch ASAP before they get sent out.
What is will you see as much variability within the same site when you measure the same sample, like a month?
We have some of that data. We have stability studies that we had done just on a master batch. So we had done some reproducibility between the different sets of dime bag samples, and also longitudinally in a freezer versus in my desk drawer to show that it was stable and making those same measurements over a couple years, because these were generated 2 years ago, almost exactly.
So one thing is that we also are going to hear from the iPRG.
Yes, we're not actually the iPRG, but we kind of focused more on the informatics.
Let's do one more question, then you can talk in.
So, yeah, just a technical question. Same amount on top for everybody?
Presumably, I can't control how much they actually loaded.
No, we didn't tell them anything.
No.
They were left to their own devices. And they analyzed data.
Right, so now just giving my 2 cents for it. I think if you're running any kind of standard, you should run a low amount of that standard, not more than 40 or 50 nanograms on the column. I think that you have to allow the column and the chromatography to work optimally, and it does that with a low sample load, and it's more reproducible if you always run a low sample load.
I don't
Not as bad as you might have thought.
Yeah,
It looks weird, but not bad.
I don't think that that's the spectrum counting data.
Are you sure?
Yeah.
Okay.
Yeah, I don't think it's spectrum counting data. But there was some spectrum counting data that was generated that mostly got the same ratios as well.
From a very good lab.
Yeah,
Oh, that was, yeah.
that was from Magnus's lab.
I think that was the last entry, 12. I have the unblinded version.
Why don't you say we're working on a manuscript?
We're working on a manuscript, but yes, the goal here was not necessarily supposed to be the informatics. It's supposed to be a product that you can purchase, and now you can take this to your own group and test this. Test your software, test your hardware, and break it. See where it breaks. That's the goal.
If you want to use it as a standard, yes, you
Yep.
can inject a low amount, but if you want to see how many peptides you can get, you can inject more.
Yeah, so that was the goal. We've developed this physical sample that clearly is addressing something that we don't have currently in the community that we can now use across whatever resources, across different operators, across different vendor hardware. In all of these different ways we can slice and dice the data. But importantly, it's not just people buying their own and mixing their own at home, which is not a standardized way for us to be doing this. And that's what everybody's been doing so far, kind of making their own homebrew versions of this. That could be easier or harder to analyze depending on what their intrinsic motivations are. All right. Very good discussion. I think maybe a little longer than. Hopefully we didn't take too much from you.
Yeah, I will.
While Pratik is loading the slide, again, I encourage you, if you have some interest in joining one of the research groups, we are going to be merging the sPRG and iPRG. It doesn't mean that we won't be working on standards or some other project, but we would love to have new members and encourage you, even if you're a new ABRF member, it doesn't matter. If you're looking for your interest, and you don't have to be a high-end expert in that area. You can look for the user input too, just people participating. And no, you don't have to do a lot of work. There'll be usually a monthly meeting by virtual at a reasonable time, and you would join in and give your input. So please let us know if you would like to join.
All right, so hello, my name is Pratik Jagtap. I work here very close by University of Minnesota. I'm part of the Proteome Informatics Research Group. And most of this work that I'm going to talk about today is basically done by Michael Hoopman, who's the co-chair of the iPRG group, as well as Sue and Victoria Dorfer. So I'm basically here to present most of the data. The first slide here basically shows some information about the Informatics Research Group. And if you're interested, as Sue said, please scan the QR code and you'll get more information about the Informatics Research Group. You can, of course, use the link as well, but I think the QR code is easier to get to the website. So the iPRG, that's the mission statement that we have here. We basically are a group of ABRF members who work in the area of bioinformatics, and the idea is to provide accurate and comprehensive analysis of proteomics data, especially new data sets that, emerging data sets that come into, that are being available. And since 2009, there have been quite a few projects. The latest of these being in 2020, we did one on metaproteomics, which has been published in the JBT. And then 2022, we had a cloud computing workshop, or at least 3 different platforms that we use for cloud computing. And the tutorial material is available on YouTube. The one that I'm going to be talking about is cross-linking study, so protein cross-linking. And the idea here was to evaluate various methods that are available, or or bioinformatic methods that are available for protein cross-linking. And protein cross-linking, as you might be aware, is basically used to study protein interactions. And in general, what you do is you start with with proteins of your choice, use cross-linking reagents, and then you digest them with trypsin, like you would do for a normal proteomic experiment, and then perform mass spectrometry, and then perform database search of the proteins that are present in your sample. At the end, you end up doing some analysis to look at structural restraints or interaction maps. And the specific aims of this cross-linking study was to provide the participants with cross-linking data sets. So we provided them with cross-linking data sets using cleavable cross-linkers. And we also provided them with some example workflows and tutorials so that people who are new to cross-linking can use those, can look at these tutorials or workflows, and then use them on the samples that were provided. And the idea was also to collect these data sets that were provided by the analyzed data sets that were provided by the users, and then highlight the challenges that are associated with analysis of cross-linking data. And then eventually, whatever that we learned from this data that we received, the idea was to educate the community so that there could be some kind of common practices that could be suggested to the for computational analysis. So, yeah.
Are these cleaved chemically or in the mass spectrometer?
These are cleaved in the mass spectrometer. Yeah, so that's the one. So as you can see in that first left side, yeah, we provided at least either 2 protein mixture or 3 protein mixture. So this cross-linking study was in 2 phases. The first phase was providing with 2 protein mixture. And in that we use this cleavable reagent, DSSO, and then the MS2 analysis was performed. The participants basically received the data in raw or MZML format, a protein FASTA file with those protein sequences, and also information about the cross-linker mass and structure. And then there were the 3 tutorials from 3 different software packages that were provided to the users for MS2-based analysis. And then we also provided them with a template form for results so that it would be easier to compare the outputs that came out of these. And then what we received from participants was a list of proteins and the positions, the cross-linked positions between them, also statistical probability associated with each link interaction, and then also information about the software that was used along with the parameters that they used for the analysis. So once we had that information, this study basically was in 2 parts. As I said, the part 1 was we provided them with these tutorials for Mascot, for Kojak, and also from MS Anika. These are 3 different platforms or 3 different tutorials that have been developed from 3 different platforms. And this actually helped some of the researchers who are new to cross-linking to kind of use these and then so that they could apply it for the analysis. So what they did was they looked at these tutorials and then they applied this, whatever that they learned from this part 1 on this data. And again, there was no prior knowledge of cross-linking that was required to participate. So there we have some experts, but we also have some people who are new to cross-linking studies. So this is the kind of the first overview of what we got from the analysis. So what is shown here is basically in blue bars are these cross-linked spectra matches, which is almost like peptide spectral matches, but here we are just looking at proteins that are peptides coming out of proteins that are cross-linked. And cross-links are, so these are more like PSMs, while the cross-links are almost like peptides, wherein you're looking at each unique pair of these cross-linked peptides. And you can see that there is a lot of variability in number of detections. And this came from 7 different participants. And I mean, luckily for us, all of them used a different pipeline. So this is a good way of basically going back and finding out how different are these results that we get from these, what are the different validation algorithms that we used, or even search algorithms that we used. And then there's an opportunity to compare results at different error thresholds. And as you can see, there is a noticeable diversity in the identification statistics that we got here. So this is a slightly, I'll go through this slide as well as I can. So Michael Hoopman made this slide. So what is basically shown here is these two proteins, the protein A, which is a FOXA1 protein, which is shown on the left side. This is shown in red. The one on the right is protein B, which is androgen receptor protein, which was the other protein that was used. If you look at the outermost circle here, which is also shown here, these are the amino acids positions for each of these, for each of the protein here. And in the blue line here, for example, for this protein, the dashed white lines are basically the lysine, the lysine position, which is where the cross-linking would take place. And as you can see, in some cases, that cross-link was not detected, while in some cases, that cross-link was detected. The blue lines basically here show interactions within the protein, and the red lines show interactions, within protein A, and the blue lines are within protein B. The purple lines are basically interactions between those two proteins, and these are of most interest that we are going to investigate. And here again, one more detail: the lines here, as well as the the length of these lines, as well as the darkness of lines, basically represents the number of participants who could detect these crosslinks. So it's a pretty informative diagram here, and we plan to use this to understand how well each of these labs did. But one of the things that came out of this is that despite the different number of software, as well as different number of PSMs, not PSMs, but the crosslink matches that we got from each of these labs, there was a core set of crosslinks that were identified among all participants. So as you can see here, all these 4 crosslinks were basically detected by all of the labs, as well as the ones that have been highlighted here, the ones that have been shown in red. And the differences that you could see, the ones that you're seeing, which are slightly light, that were detected only by a few of them, could be because of confidence thresholds or accuracy of error calculation, or even some manual curation that was done. One of the things that came out of this was that, at least for protein A, only one site was detected that that linked to 4 different sites for protein B. So we are going to investigate whether, so again, this was the first phase of the data that we got, and we're going to investigate further in terms of how accurate are these detections. Most probably, since all labs detected them, it's quite possible that these were accurate, but we are going to go deeper into the data and look into this slightly more closely. So just to kind of conclude, we invited participants to take part in a 2-part study, and what I've basically presented here was just with the 2-protein system. There is also data that would be available for 3-protein system. For the part 1, 7 participants sent us 7 different pipelines, which was good because I think some of them were really new to this analysis, and they wanted to try different software pipelines. Some of them used their own pipelines that they had used, and which also led to a diverse set of results. But despite this diversity, there was a core set of crosslinks, as I showed earlier, that everyone identified. So that was good to see. And as as I said earlier, a single site of protein A interacted with multiple sites of protein B, and then additional interactions that were detected will need to be assessed for their confidence, as well as error threshold differences, or the sensitivity of the analytical pipeline that was used. So there are quite a few features of factors we'll have to look into to kind of find out how good are these matches. We also plan to use phase 2 study, which will also have 3 proteins, and hopefully understand some more details about cross-linking. But again, I think where we are right now in terms of with the goals that we had for the cross-linking study, we are pretty well placed in terms of the number of participants and the variety of data that we got, as well as we also hope that we should be able to educate researchers based on the analysis that we'll perform in terms of how cross-linking data can be analyzed. So yeah, that's kind of where we are at this stage with the iPRG study. And I don't know if there are any questions. I'll be happy to answer them.
I had a question about the Hoopman figure.
Yeah.
Well, that's what we'll call it from here on.
Yeah. All right.
Yeah, fair. So the purple lines are the crosslinks between the two. There are other crosslinks between the two that are identified by fewer participants. How many crosslinks were identified, say, by the majority of participants? Not every participant, the majority of participants. Were there more that crossed between the two proteins?
I don't know. I haven't looked at the data, but definitely something we can look into. What have been highlighted here is something that was detected by all of the participants, but you're right, there could be some which could be done by 5 of them, but not all 7 of them, and that would be worth looking into.
Wrote something to draw that, and we have to figure the best way to present it.
So it certainly seemed like some of the participants identified far fewer peptides than other participants.
Right.
And it might send a more clear message about sort of like what the majority are doing to highlight the majority rather than just the maximum, right? So like
Yeah.
6 and 4 and 5 were
Yeah.
much lower than the other.
Well, what we need to factor in is first off, look at their method and see if some use of more stringent criteria. So there's a whole lot more evaluation of the data that needs to be done. And what we did, and I'm pretty sure the data is still available, at least on the pipelines, but Mascot, Matrix Science made this available on their public site. So people who want to try it still can access the data and try that for themselves. And Kojak is available through the ISB.
Yeah.
I'm not sure about Anika.
If your question is whether two proteins interact, what do you need to see in the cross-linking data to give you enough confidence to say there's a good likelihood that they do? Do you need one cross-link? Do you need the 4 cross-links that you show on everybody's hats?
So
Or what do you need to have confidence that two proteins do
this is
interact?
really a different question.
It depends upon the proteins,
This is
right?
not asking globally what proteins are interacting. This is a study where someone is looking at this complex for cryo-EM or other methods. And there are parts of the protein that are not accessible by the microscopy. And in fact, this is a real data set that I did for somebody. And the mass spectrometry provided answers that she could not get, and it was enough to then make a really good paper. So it's not asking globally what are interacting. This is not the method I would use for interactomics. But this is the same, what parts are interacting. And what's not shown on that diagram, which is really incredibly important to the structural biologists, is there are also what they call monolinks. So links that reacted to a site but then were hydrolyzed off, so they're not connected to another protein. But they actually show you solid accessible regions that are very informative, because the one on the left had many fewer and was definitely protected by the complex.
Is the structure of this complex known? Like, is there a
Now,
by
Androgen receptor is
Yeah.
pretty. I thought this was awesome because so I do transcription factors, so this is literally like all I look at.
look up Rossman.
I was looking up the AlphaFold 2. Androgen receptor has like some subdomain that's highly structured, but some of these could just be because all of the floppy unstructured bits you might happen to capture once or twice and measure that once or twice. So I see some of this like the super strong 4 crosslinks there
some help, and so I said, sure, I'll try it, and I really used just standard MS technology. I think there's some ways we could enhance the data acquisition, but then I did it with Mascot because that's what I had available, and I was amazed at how valuable, and she was just jumping for joy when she got the data set. So I thought that was something beyond access to a standard core laboratory, but in fact it's not. Now, the key is you need the investigator who knows how to make the samples. That is not trivial. You need an expert. You don't just have somebody throw together two. Well, for example, if you're doing global interactomics, that's something much.
No, I'm not talking about global,
Yeah.
I'm talking about a question about specific proteins that are in.
But they already know that these proteins interact. This particular question is not asking which proteins. But now, in the second part of the study, there are 3 proteins, and it was not known how those 3 might interact. But they had also cryo-EM data that they needed to corroborate. So, again, this was going in with recombinant proteins, not cell lysate.
Great. We can talk. We can talk later about a similar to this project.
Any questions?
Yeah. If
Anybody interested in signing up? Well, I hope so. You can come up or you can email us. And you don't have to be the ultimate expert in these technologies. We want all levels of people participating because, I'll tell you, I've been part of the iPRG, and the last time I wrote code was the 1980s, but I want to represent the actual user who uses commercial software and want to see that these studies are amenable to local users.
I think this is very encouraging data because cross-linking has been traditionally wicked and knowingly hard.
I was amazed that it corroborated, it meshed with the cryo-EM data, but also provided information she couldn't get from the cryo-EM data.
No, it's very, very encouraging.
And what I can tell you that,
We're moving forward.
yes, well, no, I was excited, but what was neat that we could do with it is the output from any of the softwares can then go into some other publicly available. I use what some people call ZView, which is a very easy way to put it in that makes these circle plots, if you want, or line ones, and gives you metrics on the reliability and such. So, like I said, any lab can now do it with software, and you don't have to have people writing code.
So, Sue, do you want to discuss about how RGs can work together right now, or what time do we have? Some more time because
It's 5:30, but
we have. I was wondering if I think one of the parts of the agenda was to discuss if there are any projects we can. So maybe.
that's true.
Yeah.
We're pretty much out of time. I think we're supposed to finish.
In 15 minutes?
Oh, we have 15?
Yes.
Okay.
So we have time to talk.
Okay. So what we're also looking for ideas is new projects and projects that can work either between the Proteomics Research Group or some of the other research groups that you may be involved in and interested in. So we would very much appreciate your input. Any ideas now? Anybody want to talk about a project you have in mind?
So I had a question for the first project, right? I mean, you have the data now, which you obviously have the capability of.
Look at those,
In fact, we could probably pull one of these, or pull this one and.
Okay, so the question was for the project that you have for the 3-organism mix sample, you obviously have the capability of doing the bioinformatic analysis, but do you think reaching out to iPRG and getting some of their ideas would be one of the, and do you want to involve the iPRG in doing some of the analysis, or?
I think we would be very happy to have the iPRG get interested in trying to interpret the data. I think that we are planning to set up a simple pipeline to demonstrate essentially that all the results that the different labs are producing is pretty good, right? That no matter if they did DDA or DIA or whatever, that they all kind of get the right answer. And that's kind of where we're planning to leave it off. If you want to take a data set and make it available to people, we could get approval, or we could select out of people that we know for you to, as the iPRG, to make one of those data sets available for further reanalysis, for further investigation into something that I think would be really interesting is the aspects of protein homology that this data set give us the ability to ask questions about without actually, like, we didn't try to probe into that, but what does it mean to see a sequence variation in a peptide? Like, now we actually have a nice clean data set to be able to tackle questions like that, that the iPRG could absolutely try to run, right? We want to design this so that it had legs beyond sort of what we envisioned.
I think it's a great idea, the iPRG, to take it up, because it does have so much information that can be looked at from many different directions.
And
All the data's already in print, I could just send you the link.
sure.
We do want to make sure that whoever does it has agreement to have it be part of this next
You
set.
can take
But there's plenty of sets that we could move in that direction.
Sure, yeah.
But I think it would be worth it to take more than one set,
Sure.
because the whole idea is to compare and see. Would any of you be interested in getting a sample and trying?
So how did you analyze the data? I missed that part. Did the labs themselves analyze it?
Yeah.
That's part of the,
That's part of the challenge.
part of the question.
Right.
But now the thought is to take the data and reanalyze it.
Yeah, do you have,
Yeah.
So we still
You
have
guys wanna
sample,
do
and we
that.
have not told you what the ratios are. So that can also be if you want to take them some sample.
I don't have it all the way.
Yeah.
I do,
You
I
don't
have
have
one
it
of the
first?
tubes that somehow they like this out.
I mean, yeah.
But it's only one,
I got a random tube in my bag, you know.
Yeah, that's not gonna quite do it.
yeah.
That's a mama purse there.
yeah, I want
That's
to
a
walk through the
goldfish too, if anybody needs a sniff.
my says
I mean, I can tell you that we did it, we did do it by DIA, but we just used the standard method. And I mean, it's interesting what Paul brought up about looking at it as a standard. We didn't view it as analyzing it as a standard that we would for testing system suitability and QA. We did it to try and investigate contents. So we injected 2 micrograms probably, which is what we do, because my philosophy is if you have plenty of sample, use it. As long as you're not saturating your system, why say, oh man, I can get this data only at 10 nanograms, but you can get a lot better data higher. But for a standard, absolutely, you want it to be at that lower level. So I hadn't thought of using it for a standard in that sense.
Yet you're talking about it as a standard, so that's what I thought.
Well, a quantitative standard, well, but again, that would be fascinating for people to try, to see if I inject it at this level, this is what I get, and if I, how much lower can I go down and get ratios, and do the ratios get better, and what do I lose when I drop off lower abundance, does that make the result better? So I think that's a great study that each individual lab could do.
Also, one more question, like, are these representative of various mass spec, not only vendors, but also generations of mass specs that have been used? Does it have
Yeah,
Tim
there's no
stuff?
astrals,
There's no astrals yet, okay.
thermo, I gave them samples and
Okay.
never came back, so.
Do you have,
But there's a variety of tops, I believe. There's definitely some Tim's
subtleties maybe that,
Potential, yeah, sources of variation.
Yeah,
so that's why it would be really neat to look into it in depth.
yeah, yeah. Was any of this XenoSWAT data, any SciAx instruments, or?
I don't know the answer to that.
Okay. Ben Garcia has one, right? I think. I mean, I'm sure there are other labs which have. But
We're losing you.
yeah. So Ben Garcia has one XenoSWAT instrument, but I'm sure there are other labs who have it. Maybe getting that. Yeah.
Any other comments? You think about projects. Go home after what you've heard today, and maybe you have an idea, something that's come up in your lab, something that is a challenge for you, and it would be neat to have input from others and have others try the same thing.
One more question. So, kind of linking your yesterday's talk, can these data sets also be run on non-mass spec methods like the Nautilus? And
Not for affinity-based experiments because
yeah,
they're
yeah.
peptides.
Oh, right.
Very digested. We still have the original protein,
Mhm.
the not digested protein. I have
But
some
not
stocks
mixed.
of that,
Not mixed.
but it's not mixed.
Okay. Okay.
But as part of commercialization, our commercialization plan, which is, to be clear, we're not making any money out of this. What we're trying to do is get someone else to manufacture it for us.
And not to have it as a,
It's a positive. We have someone that we're working with so far, but they're problematic right
yeah,
now.
yeah.
So we can discuss that after. But the idea is that we have a recipe now for what this thing is to give it so that other people can manufacture it from things that we just were able to purchase. So all of the standards that went into this were NIST standards. So we were able to kind of mix them at an appropriate level and do all of the preps and do everything the way we had it standardized. And then someone else could potentially follow that as well. And our plan is for the commercialization people to be able to do that too.
But not an expensive standard, something that's realistic to keep buying. But hopefully that there is enough of the initial source so that it's not something you're going to look at batch variation in the future. And at least from the initial testing that Lindsay did, there was good stability over a reasonable amount of time. Under a lot of conditions, and stay
Some intentional and
might
some not.
expensive.
I mean, all of the samples have flown multiple times now.
It's here and back to Seattle.
Right.
And out at room temperature.
Oh, yeah. The fish is pretty gross. That's traveled all over America and not always refrigerated.
The peptides are fine though.
The peptides are fine, it just smells.
You don't see any differences, right? For peptides based on,
No, the peptides are great. The original NIST fish is just
Rancid.
literally fish. And it is it was in someone else's hands before it got to mine in 2022. Fish doesn't last that long.
So there's a very interesting piece in Cook's Illustrated. I love cooking, but I love cooking as a chemist. And Cook's Illustrated always has why this recipe works. And read up about trimethylamine N oxide that's on the surface of fish. Because my mother, who was definitely not a scientist, always soaked her fish in milk before she cooked it. And she did things because that's the way she did it. And it turns out that there's an absolute reason that on the surface there's this trimethylamine N oxide. And as the bacteria work on it, it becomes trimethylamine, which you know how that smells. Well, if you soak it in milk, what does it have a lot of? Casein. What does casein have? A lot of phosphosites. And the phosphos grab the trimethylamine, you wash the milk out, and you wash the fishy smell away. And it really works.
Skin or skinned fish? Skin
Well, it's
or fillets?
the skin, but I can tell you that soaking the skinned fish in milk also does it. So there must be some of it that is on the inside. And it works.
I'll remember that.
Yes. That's just a little extra. Don't use the milk after that.
maybe a cat would like it.
But in that same Cook's Illustrated article, I guess we're getting recorded, but that's okay. I'm giving attribution to Cook's Illustrated. It talks about